My name is Layla Auter, and my project was mitigating harmful algal bloom contributing nutrients, Year 2, comparing the effectiveness of the amino acid derivative cysteine methyl ester alongside aluminum sulfate to reduce aluminum and soluble reactive phosphorus in water treatment in both hypoxic and noxic conditions. Harmful algal blooms, or HABs, are an escalated state of algal blooms that have appeared in Gulf coastal waters for nearly a century, primarily from eutrophication or the presence of nutrient overload as a cause of pollution. One component of eutrophication is the presence of high soluble reactive phosphorus, or SRP, and lower dissolved oxygen, or DO, concentrations, the latter of which can be further escalated with the introduction of HABs into water systems. One type of HAB that has been particularly troublesome is Corynea brevis, or the red tide. Corynea brevis releases a potent neurotoxin called brevetoxin upon the death of its algal cells, which targets neurons in organisms and causes the neurons to constantly fire off, leading to symptoms like paralysis or even death when the toxin is ingested or inhaled, seen with fish kills around in our area. Recently, it was discovered that the amino acid derivative cysteine methyl ester, or CME, has the ability to neutralize the threat of brevetoxins by forming an enzyme-substrate complex, and has the ability to also remove or reduce heavy metal ions. One of the most popular methods for soluble reactive phosphate reduction treatment in the surface water is combining substances with a metal coagulant, the most effective and cost-effective being aluminum sulfate, or alum. Alum reduces SRPs through the production of a flock, and in the process releases aluminum ions that in high concentrations can be toxic. Thus, alum treatments are limited to application in small doses. In the previous year, the predecessor to this project found that a combined treatment of CME and alum has the capacity to successfully reduce total aluminum, or TAL, and SRP to under their regulated accepted standards in a simulated coastal water setting. This study intends to demonstrate the combination of CME and alum as an effective distribution method for the neurotoxin neutralizer and will prove to be a successful and cost-effective method of reducing SRPs and aluminum ions released from the alum portion of the treatment under both anoxic and oxic water conditions. And it should be noted that from here on, in the pictures, hypoxic trials will be labeled as anoxic. See limitations later on. To test the effectiveness of the treatment under oxic and hypoxic conditions, first a standard solution, or coastal water stimulant, needed to be created. To do so, 5 gallons of dechlorinated water was spiked with potassium phosphate in order to reach an SRP concentration of 0.5 parts per million. From there, 7 beakers were filled with 1 liter of the standard solution, 1 for the control, and 3 each for the oxic and hypoxic. Using either nitrogen or ambient gas and a DO meter, the beakers were manipulated to reach their given conditions. Once the beakers were set up, one by one, they were added to a stir plate and mixed at 60 RPM for 5 minutes upon the addition of 0.25 grams of CME and 0.07 of grams of alum, concentrations determined as the most effective dosage for this condition in the predecessor project. After 5 minutes were up, the beakers were removed from the stir plate and allowed to sit undisturbed until a flock was produced and allowed to settle. Once the beakers had produced a flock, samples were taken from the supernatant, or the top of the beaker furthest away from the flock. The samples were then run through TAL and SRP tests in order to determine their concentrations using a colorimeter. In order to measure the effectiveness of the treatments, the oxic, hypoxic, and control trials were compared to the previous results of trials, including an alum-exclusive treatment and data collected from the standard solution and distilled as a baseline. The yellow area displayed on the graph represents the target reduction area for aluminum concentrations in parts per million, or ppm, as established by the EPA, the limit being 0.2 ppm. The standard solution had a baseline TAL concentration of 0 ppm in order to best simulate common current coastal waters. The alum exclusive treatment had an average of 0.3 ppm TAL concentration, nearly 33% above the maximum allowed concentration limit. All dual treatments were able to reduce the TAL concentration below the maximum allowed limit, with the control being the most successful, followed by the oxic and hypoxic trials. The yellow area displayed in this graph represents the target reduction area for SRP concentrations in ppm, as established by the EPA, with the limit being 0.05 ppm. The standard solution had a baseline SRP concentration of 0.5 ppm in order to best simulate common current coastal waters. The alum exclusive treatment had an average of 0.01 ppm SRP concentration below the maximum allowed concentration limit. All dual treatment trials were able to reduce the SRP concentration below the maximum allowed limit, with disruptions not being significant enough to push the SRP concentrations outside of that allowed limit. In this case, the oxic and hypoxic trials had similar results in reduction and actually showed an improvement in the success of the treatment's ability to reduce SRPs. Statistics run on the TAL and SRP concentrations both showed extremely low p-values of about 2.658 times 10 to the negative 10th, which indicates that the results received have a high probability of being replicable and the data was statistically significant. The results showed that the hypothesis was supported by the data. Under both oxic and hypoxic conditions, the treatment was able to reduce TAL and SRP below their target values. The oxic trials disrupted the reduction of TAL by approximately 2.3%, and the hypoxic trials disrupted the reduction by 57.47%. This shows that when it comes to TAL reduction, it's optimal to use the treatment under oxic conditions rather than hypoxic or regular. Both the oxic and hypoxic trials showed an increase in SRP reduction compared to the control. The oxic trials displayed an approximate 0.72% increase in reduction capabilities, and the hypoxic trials displayed a 2.13% increase. One notable observation that occurred throughout the experimentation was the increase in floc settlement time the hypoxic trials experienced. The oxic and control trials were able to see floc production and settlement within 10 to 20 minutes of addition of the mixture. However, it took upwards of 45 to 60 minutes for the treatment to produce and settle the floc under the hypoxic conditions. While this didn't appear to affect the effectiveness of the treatment, this makes the application of the treatment under these conditions less appealing. These results indicate that the treatment would be most successful under all dissolved oxygen concentrations. However, the best method for application would have to be under oxic. In particular, one real-life application area the treatment could be used under is muck dewatering. The process of muck dredging and dewatering is a method currently being used to combat eutrophication. Muck dredging involves removing large quantities of muck from bodies of water and allowing the muck to be removed and then filtered and returned into the system. In the dewatering process, it would be a viable location for the application of the treatment, as you could take the water retrieved from the muck, place it in an area to be aerated, and then treat with CME and alum, then return to the system. This way, the water would help to distribute the brevetoxin neutralizer, reduce SRP concentration in water, which would eliminate one of the largest contributions to algal blooms, and reduce TAL concentration that would be spiked by the addition of the alum in the treatment. During the testing process, there were some limitations that restricted the intended goals. For example, authentic coastal water was not allowed to be collected, so a standard solution was required to be made. This process simplified the testing process and any possible conditions that could affect the results, but in that way the treatment has yet to be tested under real environmental factors. In addition, true anoxic conditions or a total lack of DO was unable to be achieved, so the anoxic trials were changed to hypoxic, although labeling remained the same. Another obstacle that was encountered was the lack of nitrogen gas near the beginning of testing before trials were conducted, which extended testing time by a month in order to get a new tank. For testing itself, it was assumed that any changes in TAL or SRP content was the result of addition of CME or alum. In the future, the project could be extended into areas to investigate the time the residual effects of the treatment last and how to combat the disruption of SRP reduction on account of the CME. Another area for study would be investigating the effects of treatment on local wildlife, which was something that had been initially planned to be studied this year, but because of regulations, it was unable to be pursued. Thank you for your time.